Welcome back to the deep dive. You know, if you've ever had blood drawn or been given an antibiotic, you've directly benefited from the technology we're getting into today, the microscope.
Absolutely. It's fundamental.
Yeah, and we're not just talking about looking at tiny things. These instruments can literally be life or death. Think back to 2001, the anthrax bioterrorism attacks.
A really chilling example, and it absolutely highlighted how critical basic micro tools are right then and there.
Totally. Remember that photojournalist in Florida?
Yeah.
Suddenly got really sick, confused, disoriented? They did a spinal tap, sent the cerebrospinal fluid, the CSF, off to the lab.
And what happened next was, well, it was textbook rapid diagnosis. Saved critical time.
So what did they see?
A simple gram stain. That quick look at the CSF showed these really large, distinctive gram-positive rods, kind of in chains.
Just from looking?
Just from looking. The morphology, the stain reaction, it immediately screamed inhalation anthrax, a disease, incredibly rare, hadn't been seen in the U.S. for years.
Wow. So that visual diagnosis allowed doctors to start the right treatment, like, immediately?
Exactly, based purely on what they saw under the microscope.
Okay, that really sets the stage. So our mission today is basically to give you the fast track, the shortcut, to understanding these essential tools. We're diving deep into Chapter 2, microscopy.
Yep. We'll cover how different technologies let us see the microbial world, overcoming the limits of our own eyes, from basic shapes all the way down to, well, almost atoms.
We're looking at light, lenses, resolution, how you prep samples, staining, and then the really high-tech stuff.
Right. And to really get microscopes, you first have to get light. And how lenses work, it all comes down to refraction.
Refraction, that's light bending, right? When it goes from air into glass, say.
Exactly. It bends because light slows down when it enters a denser medium like glass.
Yeah.
How much it slows down is measured by the refractive index.
Yeah,
And the difference in refractive index between two materials like air and glass determines how much the light bends and in which direction. Think of a prism splitting light.
Right, right. So a lens is basically like a carefully shaped collection of prisms.
Pretty much, yeah. A convex lens, the kind that bulges out, bends parallel light rays so they converge at a single focal point, F.
And the distance from the lens center to that point is the focal length.
Correct. And here's the key part for magnification. A shorter focal length means more magnification.
Ah, okay. So shorter focal length, higher power. That makes sense why the high-power lenses are so small and close to the slide.
Precisely. They need that very short focal length to magnify things enough for us to see, far beyond what our eyes can focus on up close.
All right, so let's build up to the actual microscope you see in labs, the compound microscope. Why compound?
Because it uses two main sets of lenses. First, the objective lens. That's the one closest to the specimen. It creates the first magnified image.
Sure.
And there's the ocular lens, or the eyepiece, which you look through. That magnifies the image from the objective even further.
Got it. And the total magnification to just multiplying the two. Like a 45x objective and a 10x eyepiece gives you 450x total.
Simple as that. But, and this is a huge but, magnification on its own is kind of useless if the image is just a big blur. What you really need is resolution.
Ah, resolution. That's the critical difference. So magnification makes it bigger, but resolution makes it clear, being able to tell two tiny things apart.
Exactly. It's the ability to distinguish two small objects that are really close together. And there's a fundamental physical limit here we're always fighting against with light microscopes.
What's that limit?
For a standard bright field microscope, the best resolution you can possibly get is about 0.2 micrometers, often written as 0.2 micrometers. That's roughly the size of a very small bacterium. You simply cannot resolve anything smaller than that with visible light.
0.2 micrometers. Wow. What determines that limit? Is it the lenses?
It's physics. It comes down to two main things, basically described by the Abbe equation. First, the wavelength of the light you're using. Shorter wavelengths give better resolution.
So blue light would be slightly better than red light.
Yep, a little bit. The second, and maybe more practically important factor, is the numerical aperture, NA, of the objective lens.
Numerical aperture, NA. What does that actually measure?
Think of it as the light-gathering ability of the lens. It's defined by n times sin theta, where n is the refractive index of the medium between the lens and the specimen.
Like air.
Usually air, yes. And theta is half the angle of the cone of light that can enter the objective lens. A higher NA means the lens gathers more light and, crucially, gives you better resolution.
Okay, so we want a high NA. How do we boost it? Air's refractive index is pretty fixed, right? Around 1.0.
Exactly. Air limits the Na to about 0.995 practically. To get beyond 1.0 and significantly improve resolution for those high-power objectives, we need to replace the air.
With?
Immersion oil.
The oil you always see used with the 100x objective.
That's the one. Immersion oil has a refractive index very close to that of glass, about 1.5.
So what does replacing the air with oil do?
When light rays pass from the glass slide into air, many of the rays coming off at wider angles are bent or refracted, so much that they miss the objective lens entirely. They're lost.
Okay.
But if you put oil there, which has the same refractive index as the glass, those light rays don't bend away. They travel straight into the objective lens.
So you capture more light, especially those wide-angle rays.
Precisely. You capture more light rays, which effectively increases the angle theta, boosts the Na well above 1.0, and that's what gives you that significantly better resolution needed for the highest magnification.
Makes sense.
Yeah.
So there's a limit to useful magnification too then. You can't just keep adding stronger eyepieces.
Nope. Once you've hit the resolution limit defined by the wavelength and Na, that 0.2 micrometer limit making the image bigger just gives you a larger blur. It's called empty magnification.
Right.
So the useful limit of magnification is typically around 1,000 times the Na of the objective. For most setups, that means about 1,000x total magnification is the practical maximum for a light microscope.
Okay, so 1000x, 0.2 micrometers resolution. What if you want to see living microbes, which are often almost transparent? Staining usually kills them, right?
Correct. Staining helps, but it kills the cells. For living, unstained specimens that lack natural contrast, you need specialized types of light microscopes that manipulate the light itself.
Like what?
Well, the simplest is the dark field microscope. It uses a special condenser that blocks the central light rays, creating a hollow cone of light that hits the specimen from the sides.
So the only light that reaches the objective is light that's actually scattered by the specimen.
Exactly. Everything else misses the lens. The result is striking. You see a bright object shining against a pitch-dark background.
That sounds useful for really thin things.
Perfect for them. It's famously used to spot the thin, spiral-shaped bacterium Treponema pallidum, which causes syphilis. It's very hard to see otherwise.
Okay, dark field is good for outlines, but what if you want to see structures inside a living, unstained cell?
For that you need the cleverness of the phase contrast microscope. This is brilliant, really. It exploits tiny physical differences within the cell.
How does it work?
It works because different parts of a cell, like the nucleus or granules or endospores, have slightly different refractive indices than the watery cytoplasm around them.
Okay, so light passing through them slows down a tiny bit.
Exactly. Light passing through those denser structures gets slowed down or deviated, putting it slightly out of phase with the background light that passed straight through the cytoplasm, maybe by about a quarter of a wavelength.
Just a tiny shift. How does the microscope make that visible?
It uses a special diaphragm in the condenser, called an annulus, and a corresponding phase plate inside the objective lens. These work together to manipulate both the deviated light from the cell structures and the undeviated background light.
Manipulate how?
In the most common setup, positive phase contrast, it shifts the phase of the undeviated background light forward by another quarter wavelength.
Okay, so now the background light and the light slowed by the cell structure are half a wavelength out of phase?
Precisely. And when light waves that are half a wavelength out of phase recombine, they cancel each other out through destructive interference.
So the structure looks dark.
Exactly. The internal structures appear darker against a lighter background. This provides excellent contrast to see things like bacterial endospores, inclusions, nuclei and protists, and to study microbial motility in living cells without staining.
That's very clever. Is there anything even more advanced for unstained cells?
There is. The differential interference contrast microscope, or DIC. It's related to phase contrast, but uses a more complex system with prisms and polarized light.
Polarized light?
Yeah, it splits light into two slightly offset beams that pass through the specimen. Differences in refractive index and thickness cause interference when the beams are recombined by another prism.
And the image?
The result is quite dramatic. You get a brightly colored image that looks three-dimensional, almost like it's casting a shadow or popping out from the background. It gives fantastic structural detail on the surface and inside unstained cells.
Wow, 3D without staining.
Okay.
Moving on. Let's talk about making things glow. Fluorescence microscopes. How do they work? It's not about blocking or bending light, is it?
No, this is totally different. Fluorescence microscopy relies on light emission. Certain molecules, called fluorochromes or fluorophores, have a special property.
Which is?
They absorb light energy at one wavelength, typically shorter, higher energy light, like UV or blue light, called the excitation light. Then, almost instantly, they release that absorbed energy as light of a longer, lower energy wavelength, the emission light.
So they absorb blue light and emit, say, green or red light.
Exactly. The microscope is set up usually using epifluorescence, where the objective lens acts as both the condenser and the objective.
Illuminating from above.
Right. It shines the excitation light onto the specimen. If the specimen contains fluorochromes either naturally or because we've stained it with fluorescent dyes or maybe used fluorescent antibodies, those molecules absorb the excitation light and emit their characteristic longer wavelength light.
And filters make sure you only see the emitted light.
Correct. Special filters block the excitation light from reaching your eye or the detector, so you only see the fluorescence emission, usually as bright colors against a dark background.
This sounds incredibly powerful. What's it used for?
Oh, tons of things. Rapid identification of pathogens using immunofluorescence antibodies tagged with fluorochromes stick only to specific microbes. Ecological studies like distinguishing live versus dead cells using special dyes.
And didn't this lead to a revolution with GFP?
Absolutely. Green fluorescent protein, GFP, originally from jellyfish. Scientists figured out how to attach the gene for GFP to genes for other proteins they want to study.
So the cell makes the protein and makes it fluorescent.
Yes. You can literally watch specific proteins moving around inside a living cell, see where they go, how they function. It completely changed cell biology.
Amazing. But fluorescence must have limits too, right? What about thick samples like a chunk of tissue or a biofilm?
Yeah, that's a problem for standard fluorescence. If you have a thick 3D specimen, light emitted from structures above and below the exact focal plane you're interested in will still enter the objective lens.
Making the image blurry or murky.
Exactly. It creates out-of-focus flare. To solve this, we have the confocal scanning laser microscope, or CSLM.
Confocal. How does that get around the blur?
It uses a laser beam to excite fluorescence, but the key innovation is a tiny pinhole aperture placed in front of the detector, usually above the objective.
A pinhole. What does that do?
That pinhole is precisely aligned with the focal plane of the objective. Only light coming directly from that exact focal plane can pass through the pinhole to the detector.
Ah, so it physically blocks the out-of-focus light from above and below.
It rejects it, so you get a much sharper image of just one thin optical slice through your thick specimen.
And you can build up a 3D picture.
Yes. The laser scans across that plane, point by point, collecting the fluorescence data. Then the stage moves slightly up or down, and it scans the next plane. A computer then assembles all these optical slices.
Into a detailed three-dimensional reconstruction.
Exactly. This is incredibly valuable for studying complex 3D structures like biofilms. You can see the whole architecture, the towers, the channels, the different layers of cells.
Which helps explain why they're so tough to get rid of with antibiotics.
Absolutely. You can visualize how deep the drugs need to penetrate and why cells buried deep inside might survive.
Okay, fascinating tech. Let's shift gears slightly. We talked about manipulating light, but for basic bright field microscopy, you usually need to prepare the specimen, right? Fix it and stain it.
Generally, yes, especially for bacteria. They're small and mostly water, so they don't have much natural contrast against the background water or slide. Fixing and staining increases visibility, accentuates specific features, and helps with identification.
What does fixation actually do? And doesn't it kill the cell? Are we just looking at artifacts?
That's the constant concern, the trade-off. Fixation aims to preserve structures as close to the living state as possible, and importantly, to attach the microbes firmly to the microscope slide so they don't wash off during staining.
How do you fix them?
The most common method for bacteria and archaea is heat fixation. You smear the bacteria on the slide, let it air dry, then quickly pass it through a flame. It kills the cells and coagulates their proteins, making them stick.
But does that preserve fine details inside?
Not perfectly. It preserves overall morphology, shape, and arrangement pretty well, but it can distort delicate internal structures.
So what if you need better preservation?
Then you use chemical fixation. This is essential for looking at the fine structure of larger microbes like protists, or for preparing samples for electron microscopy. Chemicals like ethanol, acetic acid, formaldehyde, glutaraldehyde, they penetrate the cell and cross-link proteins and nucleic acids, basically locking everything in place before the cell can degrade.
Kills them just as dead, but preserves structure better.
Essentially, yes. We accept killing the cell as necessary to visualize its structure clearly.
Okay, so once it's fixed, you stain it?
Yeah.
How do dyes work? Why do they stick to cells?
Most common biological stains are organic dyes that have two key parts. A chromophore group, which is the part that actually has the color,
Okay.
and another part that allows the dye to bind to cellular components, usually through ionic interactions.
Ionic, like positive and negative charges.
Exactly. Most bacterial surfaces, DNA, and proteins carry a net negative charge at neutral pH. So basic dyes, which are positively charged, cationic, bind strongly to these negatively charged cell parts. Methylene blue, crystal violet, safranin. These are used for simple staining, just using one dye to make the cells visible so you can see their size, shape, and how they're arranged, like in pairs, chains, or clusters.
What about negative staining? That sounds backward.
It is, in a way. Instead of staining the cell itself, you stain the background. You use an acidic dye like India ink or nigrosin, which is negatively charged.
So it's repelled by the negatively charged cell surface.
Right. The dye particles deposit around the cells but don't penetrate them, so you end up seeing unstained, often bright cells against a dark background. It's great for seeing capsules or just getting accurate size measurements because there's no heat fixation involved to shrink the cells.
Okay, simple stains show shape. Negative stains show outlines. But the real power comes from differential staining, right? Procedures that actually classify organisms.
Absolutely. These use multiple stains and rely on fundamental differences in cell structure or composition to distinguish between different groups of microbes. They're diagnostic powerhouses.
And the king of differential stains has to be the Gram stain, the one used in that anthrax case.
Without a doubt. Developed by Christian Graham back in the 1880s, it's still probably the single most important staining procedure in bacteriology. It divides almost all bacteria into two major groups, gram-positive and gram-negative.
Based on their cell walls.
Entirely based on their cell wall structure.
Okay.
Let's walk through the 4 steps because understanding why it works is key.
Okay. Step 1.
Primary stain. Flood the heat-fixed smear with crystal violet. It's a basic dye, stains everything purple. Both gram-positives and gram-negatives are purple at this point.
Got it. Step 2.
Add iodine solution. Iodine acts as a mordant. It complexes with the crystal violet inside the cells, forming large crystal violet iodine CVI complexes. Cells still appear purple.
Mordant, like it fixes the dye.
Exactly. It helps trap the dye within the cell wall structure. Now for the crucial step, step 3, the decolorizer.
This is the moment of truth, you said.
This is it. Briefly rinse the slide with an alcohol or acetone alcohol mixture. What happens now depends entirely on the cell wall.
How so?
Gram-positive bacteria have a very thick peptidoglycan layer in their cell wall. The alcohol dehydrates this thick layer, shrinking the pores and trapping those large CVI complexes inside. So gram-positives resist decolorization and remain purple.
Okay. And gram-negatives?
Gram-negative bacteria have a much thinner peptidoglycan layer and an outer membrane made of lipids and lipopolysaccharide. The alcohol decolorizer readily dissolves the outer membrane lipids and passes through the thin peptidoglycan, washing the smaller CVI complexes out of the cell. They become colorless.
So the decolorizer is the differentiating step. Then what's step 4?
Counterstain. Add safranin, another basic dye, but this one is pink or red. The now colorless gram-negative cells readily take up the safranin and appear pink red. The gram-positive cells are already stained purple, so the safranin doesn't really change their color. They remain purple.
So purple means gram-positive, thick wall. Pink red means gram-negative, thin wall, outer membrane. That's incredibly informative just from a simple stain.
It's profound. It tells you about fundamental cell structure, which immediately suggests things about physiology, potential pathogenicity, and crucially, which antibiotics might be effective. Penicillins often work well against gram-positives because they target peptidoglycan synthesis, but they can't easily get through the gram-negative outer membrane.
Amazing structural insight. Are there other important differential stains?
Definitely. Acid-fast staining is critical for identifying bacteria in the genus Mycobacterium, including Mycobacterium tuberculosis, the cause of TB.
Why do they need a special stain?
Because their cell walls contain large amounts of waxy lipids called mycolic acids. These make them resist staining with ordinary dyes like crystal violet, and once stained, usually with hot carbofuction, they resist decolorization by acid alcohol, hence the name acid-fast. Other bacteria get decolorized.
So it specifically picks out mycobacteria.
Yes. Also useful are stains for specific structures. Capsule staining often uses negative staining techniques to reveal the clear or lightly stained capsule, a protective layer outside the cell wall, as a halo around the cell.
And you mentioned seeing flagella. They're usually too thin, right?
Way too thin for standard light microscopy. Flagella staining uses special techniques involving a mordant, often tannic acid, that precipitates onto the flagella, effectively coating and thickening them until they become visible. It's painstaking work.
Okay, so staining gets us a long way with light microscopy. But we still hit that zero point 0.2 micrometer resolution limit. What if we need to see viruses, or the really fine details inside a cell,
Yeah.
or even molecules?
Right. For that, zero point 0.2 micrometers just isn't good enough. Viruses are much smaller, maybe 0.02 to 0.3 micrometers. To see things at that scale and beyond, we have to leave visible light behind entirely and enter the realm of electron microscopy.
Electrons instead of light. How does that help?
The key is wavelength. Electrons accelerated in a vacuum behave like waves, but their wavelength is incredibly short, about 100,000 times shorter than visible light.
Wow. So that should give much better resolution.
Dramatically better. Electron microscopes can achieve resolutions down to about 0.5 nanometers, sometimes even better. That's almost 1,000 times better than the best light microscope. And magnifications can go way up, over 100,000x, even millions of times in some cases.
Okay, so how do they work? Do they use glass lenses?
No, glass doesn't bend electrons effectively. Instead, they use powerful electromagnets shaped like lenses to focus the electron beam. And the whole system has to be under a high vacuum because electrons are easily scattered by air molecules.
Makes sense. What's the main type?
The first type developed was the Transmission Electron Microscope, or TEM. As the name suggests, the electron beam passes through the specimen.
Through it. So the specimen has to be incredibly thin.
Extremely thin. We're talking 20 to 100 nanometers thick. You have to embed the chemically fixed specimen in plastic, then slice it using an ultramicrotome equipped with a diamond knife.
Whoa. That's intense preparation. How do you get contrast?
Biological material itself doesn't scatter electrons very much, so the thin sections are typically stained with solutions containing heavy metal salts, like lead citrate or uranyl acetate.
Heavy metals. Why?
Because heavy atoms scatter electrons much more effectively than the carbon, oxygen, hydrogen atoms that make up most of the cell. Denser regions of the cell that take up more heavy metal stain will scatter more electrons.
So they appear darker in the image.
Exactly. They are electron dense. Lighter areas are electron lucent. This creates the contrast needed to see the internal ultrastructure membranes, ribosomes, organelles, inclusions, the fine details inside the cell. TEM is all about seeing the internal cross-section.
Are there other ways to prep for TEM besides slicing?
Yes, several. Negative staining is used for small particles like viruses or protein complexes. You mix them with a heavy metal salt solution that dries around the particles, leaving them unstained but outlined by the dense background.
Like negative staining in light microscopy, but with electrons.
Precisely. There's also shadowing, where you coat the specimen with a thin layer of metal evaporated from an angle. It creates shadows that reveal surface topography, useful for things like bacterial flagella or DNA molecule.
And freeze-etching.
That's a cool one. You rapidly freeze the sample in liquid nitrogen, then fracture it under vacuum. The fracture often runs along natural lines of weakness, like the interior of membranes.
Splitting the membrane open.
Yes. Then you sublimate away some surface ice, etching, and make a metal replica of the fractured surface to view in the TEM. It gives a unique view of membrane interiors and how proteins are embedded.
Okay, so TEM is through the specimen, internal structure. What's the other main type of electron microscope?
That's the Scanning Electron Microscope, or SEM. SEM is all about the external surface of the specimen.
Scanning, so it doesn't go through.
No. A very fine, focused beam of electrons is scanned back and forth across the surface of the specimen, which is usually coated with a thin layer of metal like gold to make it conductive.
What happens when the beam hits the surface?
It knocks loose low-energy electrons from the specimen surface itself. These are called secondary electrons.
And the microscope detects those.
Yes. A detector collects these secondary electrons. The number of secondary electrons produced depends on the angle of the surface relative to the beam. Raised areas facing the detector produce more secondary electrons and appear brighter in the image. Depressions or areas angled away produce fewer and appear darker.
Creating a 3D effect.
Exactly. It generates an image with remarkable depth of field and a very realistic three-dimensional appearance of the surface topography. SEM is perfect for seeing the shapes of whole bacteria, how they attach to surfaces, the texture of biofilms, things like that.
So TEM for inside, SEM for outside. Got it. You mentioned fixation can cause artifacts earlier. Is there a way around that with electron microscopy too?
Yes, a relatively newer technique called electron cryotomography, or cryo-ET. This is really pushing the boundary.
Right, like freezing.
Exactly. Instead of chemical fixation, samples are flash frozen extremely rapidly in liquid ethane. This is so fast that water molecules don't have time to form damaging ice crystals. Instead, they form vitreous ice, like glass.
Preserving the cell in a near-native hydrated state.
That's the goal. Then, under cryogenic conditions, the frozen sample is tilted in the electron microscope, and images are taken from many different angles, a tilt series.
And then?
A powerful computer combines all these 2D projection images to computationally reconstruct a 3D volume of the intact cell or subcellular structure.
So you get high-resolution 3D structure without chemical fixation.
Precisely. It's allowing scientists to visualize molecular machines and cellular structures in situ within the context of the whole cell in a close-to-life state. It's incredibly powerful for ultrastructure studies.
Mind-blowing stuff. Okay, we've gone from light to electrons. Is there anything even more powerful? Can we see molecules, atoms?
We can. That takes us to the final category, Scanning Probe Microscopy, or SPM. These instruments don't use light or electrons in the traditional sense. They use a tiny physical probe to scan the surface.
A physical probe, like dragging a needle across the surface.
Sort of, but much, much more refined. The resolution here is astonishing. We can achieve magnifications of up to 100 million times, easily resolving individual molecules and even atoms on a surface.
100 million times. Wow. What's the main type?
The first one developed was the Scanning Tunneling Microscope, or STM. It uses an extremely sharp conductive probe, often just a single atom at the tip, brought incredibly close to the specimen surface just nanometers away.
What does it measure?
It measures a tiny electrical current called the tunneling current that flows between the probe tip and the specimen surface due to a quantum mechanical effect. This current is extremely sensitive to the distance between the tip and the surface atoms.
So as the probe scans, it moves up and down to keep the current constant.
Exactly. By recording the probe's vertical movements as it scans, a computer generates a topographical map of the surface, literally atom by atom.
Incredible. What can you use it for?
STM is amazing for visualizing the atomic structure of conductive surfaces. It's been famously used to get direct images of the double helix structure of DNA, for example.
But it only works on conductive surfaces. What about cells or proteins? They don't conduct electricity well.
Right, that's the limitation of STM. For non-conductive biological samples, especially for studying things in a more natural state, even under liquid, we use the Atomic Force Microscope, or AFM.
Atomic force. What force is it measuring?
The AFM also uses a sharp probe on a flexible cantilever, but instead of measuring current, it measures the tiny van der Waals and electrostatic forces between the probe tip atoms and the surface atoms of the sample.
How does it do that?
As the probe scans across the surface, a laser beam reflected off the back of the cantilever tracks its minuscule deflections as it interacts with the surface topography. A feedback loop moves the probe up and down to maintain a constant, very small interaction force.
And mapping those movements gives the surface image.
Yes. The huge advantage of AFM is that it works beautifully on surfaces that do not conduct electricity well, including biological membranes and proteins, even living cells under physiological conditions. Researchers have used it to watch proteins function, to see membranes assembling, really high-resolution structural biology on non-conductive samples. You can even poke the cell to measure its stiffness.
Okay, so we've really covered the spectrum, from just understanding how light bends in a lens.
All the way down to literally mapping out atoms on the surface of a cell or molecule.
It's quite a journey. And mastering these tools, understanding what they can and can't do, is absolutely fundamental for microbiologists to connect structure with function and ultimately with health and disease.
Absolutely. Each technique gives you a different piece of the puzzle, a different level of detail.
You know, reflecting on this is fascinating. We started way back with the limit of human vision, maybe 0.2 millimeters, something like that.
Yeah, about the width of a human hair.
And we've pushed microscopy technology down and down past the 0.2 micrometer limit of light, down through nanometers with electrons, right to the 0.1 nanometer scale of atoms with probe microscopy. Resolution has always been the bottleneck.
Always. Getting a clearer picture.
So, thinking ahead, since we've kind of hit the atomic scale with probes, what's the next frontier? How will future tech keep pushing? Maybe manipulating light in even cleverer ways, like those super resolution techniques that sort of cheat the old diffraction limit. Or maybe using entirely new physics. The big dream must be to see all this molecular machinery working in real time inside a living cell, without damaging it at all, right?
That's the billion-dollar question, isn't it? How do we get that dynamic, high-resolution view of life happening without interfering with it? That's what drives the next wave of innovation in imaging. A fantastic question to leave everyone thinking about.
Indeed.
Yeah.
Well, thank you for walking us through all of that, and thank you all for joining us on this deep dive into the world through the microscope.